Hello, everyone. Welcome to our BioStrand webinar, Bring Meaning to the Chaotic Space of Omics and Text. This webinar will specifically focus on the use of natural language processing, or NLP, and this in the context of omics data analysis. Today, we have three different speakers in our panel, namely Arnout Van Hyfte, he is the CCO of BioStrand, Sébastien Lemal, one of our data scientists, and myself, Dirk Van Hyfte, the CTO and co-founder of BioStrand. Just some very practical remarks in advance. If you have any questions during our presentation, you can drop this in our chat box, and at the end of the presentation, we have a Q&A session. So at BioStrand, we have our mission to build the next generation technology that makes omics data analysis truly effective. So let's suppose you are a researcher and you have sequenced some samples, and now you want to make sense of it. You want to look for new insights or create new knowledge. Well, that is a complex and time-consuming process. The data sources that you want to compare your results with are scattered into different silos. You need the help of a bioinformatician to integrate all these data sources. And that's why most pipelines have only a limited amount of data sources you can work with. Furthermore, all your scientific literature is in an unstructured format, so it takes a lot of time to go through it. So the main question is, how can we bridge the gap between the experiment, the data, and the literature? So as you know, one single experiment can generate quite a lot of hard to interpret data. For instance, if we look at the differential gene expression analysis from RNA-seq, that can generate massive amounts of data related to genes, gene groups that all influences each other. And for that, you need to augment your data with structured data sets. And that's why they, for instance, also use these biological pathways, and they call it an enrichment analysis. And at the end, of course, computers can do quite a lot, but you still need to have scientific validation based on expert knowledge. So that means you really need to look to the data with a different mindset. We need to structure the unstructured data to a structured format so that computers can work with it. For instance, the raw reads that are need to be mapped to the references. And also the scientific literature is fully unstructured. So you can only read through it, understand it by the human or by help of your computer. And that's the exact place where natural language processing can come in. So in the next part of the presentation, Sébastien will further explain how we use our NLP technology and also go through some examples. So to summarize, what is very unique with our proposition is that on the one hand, we can integrate all the sequence data coming from DNA, RNA, or amino acids with our Hyfte-based approach, but also combining with our natural language processing engine, we have, let's say, one single integrated data model, and that can accelerate R&D strategies and generate meaningful data. So Sébastien, it's up to you now.
Okay, all right. So, well, in following, I will present you a very specific use case where we use the BioStrand NLP technology to deal with our RNA-seq data and how we can output a meaningful analysis for this use case. So before going deep into the use case, I need to present a little bit, okay, what do we mean with this NLP technology? So it's all account on text mining. So the BioStrand product, which we call NLP Link, basically look up into text to retrieve meaningful words so that we can start highlighting what are the relationship between these. And what's quite different with respect to typical text mining methods, well, if we look at these methods, they are mostly based on bag-of-word models. And this means that basically we start right away from ontology or dictionaries that have been generated beforehand, which require a lot of work depending on what kind of domain you are working, because sometimes you don't have that right away available. And one of the biggest issues with this kind of text mining approach is that a word can have different meanings in different contexts. So if your AI cannot capture the context, then the meaning can be lost. And that's something that is quite problematic, specifically if you want to understand relationships between these concepts. And what the BioStrand NLP Links actually does is that it identifies right away the meaningful word groups in a domain agnostic way, which means that independently of what you are actually working on, the tools will actually pick up the right concept for you. And that's very interesting indeed, because it means that you can provide any kind of raw text, may it be scientific literature, sequence descriptions, annotation, etc. And the tool will capture the right concept right away from them. And if this is done properly, then you can build a smart index for fast document retrieval or document clustering, etc. There are different ways of proceeding then, but it means that you have basically the basic tools to do and to build more intricate models. And so, yeah, the BioStrand NLP Link is what you would call a bottom-up approach, because it captures by looking right away at the text the meaningful semantics boundaries. And so all the concepts are clearly well defined, and you don't have the affirmation issues of lost context. So to illustrate a little bit more what we mean by that, let's look at a very simple example. Let's consider that the text is just a sentence. Here, 2 patients are suffering from congestive heart failure. So here, what BioStrand NLP will do from this sentence, it will start doing a lookup on a relations and then a concept detection. And what you have at the end is that you basically have a well-defined concept. So here's the concept. The first concept is 2 patients. The relationship is are suffering from, and then the second concept is congestive heart failure. And 2 patients is a concept that is different, for example, from 1 patient. And so from that, you have a very good way of differentiating different cases where, let's say, semantic, what looks like semantic similarity can be lost. So to give you an example of what the technology does, we are now looking at what we call the knowledge portal, which is part of that technology. It's basically an UI that allows you to have a great overlook of all your ingest data. So here what we are looking first is that if we, for example, use as a keyword your query, for example, a gene symbol, in this case, you get this view where you have all the results corresponding. So this is very like a search engine, let's say, but it does a little bit more than what you would expect from a search engine. So, for example, what you have right away is that it displays right away what are the most frequent or dominant concepts that you have mined in your data. And this can be either by frequency, which means the amount of time these concepts actually appear as such in the data, or the spread. Well, for example, depending on how many documents does this term appear. So here we are looking at PubMed abstract. And so, yeah, that's basically the repartition. And it makes sense if we look at what are the top concepts coming up. And then related to your query, you have the similar entities. Similar entities are basically the entities where your words appear in a sense. And basically what's very important is that these entities should be, in principle, distinct from a conceptual point of view. For example, t-span 6 functions is not the same as t-span 6. That's not the same. And so basically, NLP link differentiate the two. Another thing that you get from this UI is the related concept, which are basically entities that occur in the same context as the explore term. And so this is quite important because then you can see that it captures the relationship between a concept and another concept. And this is basically all you have here associated to t-span 6. And something else that you can look at are the source where this concept have been retrieved, which is very important because then you can actually bring up the knowledge about the context with respect to that query. And so one of the most important aspects of this technology is that what NLP does is that it will chunk the text into different entities or tokens. That's basically the core of this technology. And what is interesting for building a model is that these different entities are related to each other in the following way. They form concept, relation, concept triples, which are the basic entities for this kind of indexing. And you may actually, if you have some knowledge of NLP, that's basically the basic structure needed, the basic units, if you want to build a knowledge graph. For example, here you have anti-inflammatory effect, which of t-span 6, which is here this effect, and this is associated to those corresponding genes, etc. And the concept relationship concept is a very important way of doing this kind of analysis. For example, in the context of differential analysis, you want to look at what is the effect of overexpression or underexpression. And so basically, if you capture here, well, either overexpression or downregulation of MCL1, you get here different kind of contexts which are different and which are important to differentiate if you want to perform your analysis. And so the main idea, as I said, is that underneath this technology, you have a very large semantic knowledge graph, which link all the different concepts all together through meaningful relationship. And so looking at that, it looks a bit messy. It's quite difficult to have an understanding on what's going on there. And the main idea is that, okay, there is a way to make sense of your experimental data by linking your experimental data right away to the different concepts that you have there. And what I'm going to show right now is an example of how you can do that and basically what you get from this kind of approach. So basically, for this analysis, the main inputs here are, well, in the case of differential gene expression analysis, you get the different gene as well as the count associated to these genes for the different case and control groups in your experiment. Okay, so that's basically an input, which is very similar to typical gene set enrichment analysis, where you basically map pathway, biological pathway to your experimental data. And here the main difference is that we are not dealing with pathway, we are dealing with a semantic graph that is extracted right away from the literature. And so in a sense, it's quite different because here we are first starting from unstructured data, which is raw text. We ingest this data with NLP link. We build a large index, and then from this, we can perform this enrichment analysis. And the typical output is a semantic subgraph where you can, for example, as attribute the statistical relevance of the concept with respect to the experiment that you performed. And this is quite important to be extract this graph. What you have in such a graph is that you have the concept, the relationship between this concept, and also the source. For example, we can have a closer look at one node, for example, which is linked to different kind of other concept node, where we see here the different relationships expressed in gray for here. And typically, once you have these relationships, you can look a little bit further into what you have ingest as abstract. Here, for example, we got an abstract from this journal that links myeloperoxidase to pro-inflammatory agents within lungs. And so here you can see right away the relationship between these different concepts like lungs tissues, inflammations, neutrophils, inflammatory marker, and myeloperoxidase. So that's basically one of the examples of what you can get from NLP link. And basically you have the tool here to start building your hypothesis, looking at a meaningful relationship in your unstructured data. So to compare BioStrand NLP link with standard tools for gene enrichments. So in the case of gene enrichments, what you use as a data to augment your analysis is that you use pathways, which are specific data structure, looking like a network, but which are simplified to gene set in order to perform the calculation. With BioStrand NLP link, right away, the data that you are using is unstructured data. So in this case, we use PubMed abstract for this demonstration, but you could use any other kind of data, like your own report, for example. Well, for the standard tools, the data that you use need to be either manually curated or automatic annotations. So if your data is manually curated, then you can have some trust with what you have. If it's automatic, a little less, but still, in the case of BioStrand NLP link, what you have is something automatic, but you always keep the context with respect to the study that you looked for. So basically, instead of keeping track of your data and looking into the literature by yourself, you basically have the machine that helps you actually looking at the specific elements that are relevant for your experience. And it also do so in a domain agnostic way. So if you are working into studying the development of cancer, or if you look into the different effects associated with fertility disease, for example, whatsoever, the BioStrand NLP link tool will actually go through that and make the data structured. Also, one of the issues with standard tool for differential gene expression analysis is that the graph structure of a single pathway is lost in this process when it's converted as a set. Whereas for BioStrand NLP link, the graph structure of the CRC triple is conserved. And so it always remains available and you don't lose that link, and also the context on which this triple has been extracted. And for the standard tools, the output, you typically have undirected network of gene set with overlap as edges, and the label for the different cluster using workload. That's typically an input that an output that you get. With BioStrand NLP link, what you get is a semantic directed graph where the node are concept and the ages relationship. So you can, just by looking at the ages between two nodes, you know right away what's the relationship between two different concepts. So to summarize a little bit, BioStrand NLP link incorporates rotate tokenization. And so from this tool, it reveals source choice, clustering, and semantic graph representation. And using this tool, you can do enrichment right away from the literature. So you perform smart indexing of scientific document with a concept relationship, concept triples, and then you can use that to build your semantic subgraph with relevance to your experimental result. And most importantly, this approach is a bottom-up approach and domain agnostic. So whatever is the document that you are looking into, the right semantic boundary will be picked. And so it does not really depend on the nature of your document, whether it is a scientific document or not. And this technology can be used to discover novel pathway, specifically in the case where, for example, there are a lot of undiscovered pathway, for example, for specific application that do not necessarily relate to humans. That can be of good interest, and also you can discover meaningful links that are not necessarily available as such in the existing pathways as well. All right, I will let Arnout take it from here.
Thanks, Sébastien. Welcome, everybody. My name is Arnout Van Hyfte. I will take the last part of the presentation into my account. And where Sébastien left off on the interpretation of RNA sequence data, I will steer a little bit more towards the, let's say, the help or the aid of having an integrated text layer in the drug target discovery process. In this process, and why it's useful to have, let's say, both the integration on the sequence level, so meaning the different omics layers, DNA, RNA, and amino acid sequences, and the power of having a way of structuring text documents and let's say processing it using our triplets in one single platform is explained a little bit here in the following slide to give you an overview of the full workflow. What does that mean? Let's say we can virtually ingest any type of sequence and any type of text document. The beauty of the system is that not only are we agnostic in the sense of I call it domains in text data, we are also agnostic in what species you would put into the system in regards to their sequence information. So all this data is, let's say, encoded into one single data framework in one integrated data model. And based on that model, you can then launch virtually any type of analysis, call it a very simple search query to check, okay, what associations do I have in regards to my query? Or as Sébastien showed you, this very complex, let's say, underlying machine learning graph where you, let's say, capture all the relationships between all the data inside your document. That could be then an ideal starting point for AI and machine learning tasks. The part that Sébastien explained in detail is, let's say, this last part of the analysis. Oh, if you could go back one slide, please, Sébastien. Yes, where we use, let's say, this NLP link to validate incoming data and to check what associations can be made based on incoming data and what can be done to, let's say, make the interpretation step, let's say, a little easier. And then indeed, if you could go to the next slide, in this next case, we zoom in a little bit on, let's say, how it could help in especially call it de-risking, let's say, your findings and in confidence building that your potential target is safe to pursue in a drug discovery process. So here we focus on self versus non-self in antibody development. And self versus non-self is to check whether your antibody is specific to a certain proteome. And in human therapeutics, well, it has to be specific to only humans rather than other organisms to, let's say, diminish potential side effects. To do so, well, the starting point is the epitope, so the binding site of the antibody, to check what cross-reactivity can be found. And that sounds rather simple because an epitope is rather short. But because we have so much residues, and you can see it on the big blue numbers, let's say the amount of amino acid combinations that you have to check, let's say, lie between 3 million and go up to 655 trillion combinations to be checked. The beauty here is that, let's say, in this first step of the process, we have our integration at the sequence level, and this helps us to, let's say, get rid of these endless combinations and do a very simple, call it lookup, based on the sequence information to then start off, let's say, the next steps of the analysis. To give you an overview of how this, let's say, analysis pipeline or workflow is structured, is on, well, where we start off at the left-hand side of the screen with any antibody, neoantigen, or pathogen. We check the Hyfte patterns, which are able to, let's say, figure out what sequences are related in either the incoming data set. We compare the Hyftes between the incoming data and the Hyftes in the database. That's the way we have indexed and integrated that sequence data. And then, well, we can virtually check any sequence that's in the data set, whether it's a DNA sequence or an amino acid sequence, anything can be taken into account. And this step alone is a big difference from current workflows and pipelines to assess self versus non-self. Because remember those, let's say, huge amounts of combinations to be checked in order to make it, let's say, computationally feasible. At this moment, it's limited to either rodent models and the human proteome, but nobody is looking, let's say, beyond this scale because it's, let's say, difficult to compute in one single process. The beauty is, because we have everything integrated, that with our machinery, but with our pipeline, that's no longer a problem. So that means that, for instance, in case of, let's say, any search, you can also take into account overlaps with bacteria, overlaps with viruses, and any other sequence you have a relevant association with. Remember again, and also in regards to what Sébastien explained, at the end of, let's say, call it this analysis where we assess whether the epitope has an overlap with the proteomes, whether it's human proteome, rodent proteomes, or any bacteria, we can do, let's say, call it a sanity check and see indeed if, based on these concepts, we can find something in the documentation, in the scientific literature, that could point to, let's say, relevant associations that can help us make sense of why we've picked it up, this association. And to make it a little bit more concrete, in the next slide, we have done this exercise based on the adalimumab antibody. So we've taken into account this antibody sequence, assessed all the relevant epitopes and checked and ranked them based on, let's say, their binding characteristics. And indeed, based on this first exercise and checking at the sequence, what proteomes and what species are associated to these epitopes, we could indeed see that there was no immediate hit with mice and rats, which is, let's say, where the current analysis stops. But there was, well, we saw a lot of associations also with bacteria. And to go a little bit further into these, let's say, these findings, the nice thing is we can also immediately verify whether there is really a relationship between this epitope and this bacteria, because we can grab those concepts and use those concepts to do a literature search. And again, in the case of this adalimumab antibody, we saw indeed the association with a certain bacteria in regards to a treatment with adalimumab, and found a lot of case reports from the clinic that pointed to very severe allergic reactions when somebody was affected by this specific microbe, or if it altered their microbiome. To summarize a little bit to what this could help and aid in the discovery process, on the next slide, please, Sébastien, is that, let's say, being able to pick up these associations can help to de-risk your antibody development. Because if in advance you can, let's say, find associations with certain bacteria, in this case it was well described in a clinical setting that it had some risk, but then you could rule out, let's say, this potential patient population, develop certain biomarkers, or do some more in-depth population stratification to make sure that you treat the right people with the right antibody. So in that case, also you can greatly improve the success rate of oncoming experiments. Next to that, because you can tailor it to the right, let's say, environment, it helps to reduce, let's say, wet lab testing, and in this specific case, could also lead to a diminished need of specific animal testing. to sum it all up, so yeah, what's the takeaway of this approach, and how can it help, is that based on, let's say, all structured data and associations, you can make data-driven decisions. It either helps to validate certain hypotheses, on the other way around, it helps to create certain hypotheses based on new relationships that you find, and that you can either explore easily then again in the literature based on other relationships that are popping up in regards to your concept. It helps in the case of, for instance, adulimumab to find new relevant associations and relations that were else missed, and that to let's say build further on the knowledge. And let's say the good thing is that, let's say, in general we have a lot of data, but in having it, let's say, presented in a structured way that can capture these associations, that helps to, let's say, put the researcher back in the center of his own data and his own experiments to check and develop further his projects. So that's, let's say, the end of this webinar. If somebody has certain questions, they can put them in the chat for the panelists to see. And if I scroll through them, I think one question that arose in regards to, call it the triplets that have been discussed, is whether we would use an ontology to capture distinct concepts from each other, or if that's the approach. I think Dirk, you are best suited to answer this question.
Yeah, well, it's indeed an interesting question, and just to make clear, the way I understood the question is if you are using an ontology to capture the distinct concepts. And so to capture the distinct concepts, we are using, call it a dictionary at the relational elements. So that means that's the way we have developed this technology, that our focus is at the relational elements. And the real advantage of that, once you can capture all the possible relations that are in a certain language, then you have for free, let's say, the semantic word boundaries. And that is a very powerful, let's say, way to analyze text, because quite a lot of approaches at this moment, they start from classical ontologies. But that means you only can find things that are well defined in your ontology. And that's like, in a certain way, finding or fishing the words or the word groups that are already in your dictionary. But there's no way to say how good your match is, because you cannot define the semantic word boundaries. And so that's our first step. We identify the semantic word boundaries. So that means if you then do afterwards a match with an ontology, you could say, okay, I have a full coverage of my word group, so I'm sure that I captured the semantic interpretation of that. Or at least if your dictionary or your ontology is not, let's say, built well enough, you can immediately see the gaps. And again, that's why this approach is really a bottom-up approach, and it's not tuned to a certain domain. And that's really important. So that means we can analyze, like, say, data coming from PubMed articles or scientific literature, but even healthcare data or electronic patient record data. And sometimes that is not well structured, that data. People are using their own, let's say, words or word groups. And so we are able to capture that. So it's very powerful. And also the fact, again, that we can integrate this with information at the sequence level. So DNA, RNA, and amino acids in one single model. It's a very powerful technology.
Yes, thank you. And that brings me to well, another related question. I think this one is aimed more towards Sébastien. Is, well, we've shown examples where, let's say, we started off from an experiment, a sequence. Could you also do the same with any predefined set of genes that is, let's say, not related to, strictly related to any, let's say, experimental data outcome, but would be a certain group of interest that you want to analyze and check?
Yes, this is quite possible. So basically, the way that this analysis is performed is that it uses, a very specific statistical model that assesses the relevance of the different concepts that you have available in the data with respect to your different gene as an input. So basically, if you provide these genes, it will look as a way to see if some concepts are indeed overrepresented and over-associated with these genes compared to all the other one. That's basically what you get. And so basically, you can also, from this kind of approach, extract the different meaningful concept and the relationship associated to them. So you will indeed have a semantic graph in such an approach. So that remains possible.
And then let's say a little side question is within these semantic graphs. So you've talked about genes and how genes are related. Potential relationship with tissues that can also be, let's say, uncovered if it's presented in literature. Is that correct?
So basically, what NLPing does is that it captures the relationship between different concepts. So if a tissues appear specifically as a concept and hold a relationship to a gene, then you can make this association. For example, in the case of some specific tissues like for example, testes, you will have some genes which are more expressed than the other. And so the other representations of the concept associated to these genes will reflect that. And so, yeah, depending on your tissue differentiations, you will have different gene expressions. And these different gene expressions can be used to detect the different kind of tissue. So that's a possibility.
Perfect. I think that is all as far as questions go. I thank you all for attending our webinar. In case you've missed the webinar and couldn't make it live, the recordings will also be available on our website to revisit. You will get them by email, and looking forward to meeting you all soon. Have a nice day. Bye.